Okay, let's dive right in with something that honestly sounds pretty counterintuitive. Imagine taking away just one component of protein, a single amino acid called cysteine, and seeing mice lose a massive 30% of their body weight. And get this, in just 1 week.
Yeah, it's pretty staggering. And what's maybe even more surprising is that it was completely reversible. Put the cysteine back in their diet and boom, the weight comes back.
It really makes you stop and think, doesn't it? I mean, a bigger effect from this one tweak than, say, cutting out entire essential amino acids in some cases.
Exactly. It just underlines how incredibly specific our metabolism can be. We usually talk about proteins, fats, carbs, right? Big categories. But this study really drills down into one of the 20 building blocks, cysteine. It's a sulfur-containing amino acid. You find it in things like eggs, meat, whey protein. Pretty common stuff.
And it's known to be important, right,
Yeah.
for making antioxidant.
Absolutely crucial for making glutathione, which is maybe our body's main antioxidant defender. And also for something called coenzyme A, or CoA. CoA is like a key helper molecule in tons of metabolic reactions, really central.
So the researchers were essentially asking, okay, why this specific amino acid? Why does taking away cysteine have such a huge effect on weight and metabolism?
Precisely. They were hoping this could point towards, maybe new, less drastic ways to tackle weight loss or metabolic diseases. And their approach was really systematic. They didn't just look at cysteine. They actually compared removing each essential and conditionally essential amino acid one by one.
To really pinpoint which one had the biggest impact.
Yeah. And they use these genetically modified mice, too, ones that literally cannot produce their own cysteine. They call them cyne mice.
Ah, right. So that really isolates the dietary rule. If they don't get it from food, they just don't have it.
Exactly. Takes away the body's backup plan.
Okay, so let's look at Figure 1. Figure 1A gives us a kind of roadmap for cysteine metabolism. It shows how cysteine is normally made through this thing called the transsulfuration pathway. You see those enzymes, CSE and CDS.
They're the key workers on that production line.
But in the special CC mice, there's that red cross showing the pathway is blocked. CSE is gone. So for them, dietary cysteine becomes absolutely essential. No ifs, ands, or buts.
And they also mentioned BSO there. That's a chemical inhibitor. It stops glutathione synthesis. That becomes relevant later on.
Right. So using these CC mice was clever because it gave them precise control. Cysteine in the diet or no cysteine.
And figures 1B and 1C, well, they basically show the money shot. That bar graph in 1B, it compares the weight loss after a week of removing different amino acids. You look at both the normal mice and the CC mice.
And the result just jumps out at you.
Totally. It's that massive, roughly 30% weight loss, but specifically in the CC mice that weren't getting any cysteine, way more weight loss than removing any other single essential amino acid they tested.
And the curves in 1C show it wasn't gradual either. It was fast. Like a steep drop-off day after day.
Yeah, really rapid.
Now, a common factor in mouse studies is temperature. Could they just have been cold, burning more energy?
Good question. They checked that in Figure 1D. They put the mice in what's called thermoneutrality, basically, a comfy 30 degrees Celsius where they don't need to work to stay warm. And the cysteine-deficient CC mice still lost all that weight.
Okay, so it's not just about fighting the cold. That points to a deeper metabolic change.
Definitely. So then you ask, well, were they just eating less? Figure 1E looks at food intake. And yes, the CC mice on the no cysteine diet did eat less, about 30% less, which, often happens when a crucial nutrient is missing. The body kind of puts the brakes on appetite.
Right, that makes sense. But here's the kicker, right? Figures 1F and 1G. That 30% drop in food intake doesn't explain the full 30% weight loss. Not even close.
Exactly. They did a control experiment where they just restricted calories in normal mice, feeding them the same reduced amount as the cysteine-deprived group ate. And those calorie-restricted mice, they only lost about half the amount of weight.
Wow. So taking away cysteine does something extra, something beyond just making them eat less. A unique metabolic hit.
That's the key takeaway there. And they show comparison data for isoleucine and valine deficiency, much smaller, unexplained weight loss. And for others like tryptophan, basically all the weight loss was just due to eating less.
So cysteine is really special in the context.
It seems so. Then in 1RI, they rule out another possibility. Maybe it wasn't the lack of cysteine itself, but a buildup of stuff before cysteine in that metabolic pathway.
Like metabolic intermediates piling up.
Yeah. So they tested mice lacking both methionine, which comes before cysteine, and cysteine, and get what?
Same rapid weight loss.
Yep.
Yeah.
Same rapid weight loss as lacking only cysteine, which strongly points the finger at the absence of cysteine itself being the main driver.
Okay, that makes sense. And figure 1I, showing the reversibility,
Yep.
that's quite striking. Stop the cysteine-free diet, give it back, and the weight bounces back up. They could even repeat the cycle.
It is remarkable and potentially really important, suggesting the changes aren't causing, irreversible damage, at least not in this time frame. It suggests a potential for control.
Okay, so figure 2 takes us into the metabolic cages, a more detailed look.
Right. Figures 2A and 2B show the setup. And again, it's only those CC mice without cysteine that show that really fast weight loss, kicking in within just 3 days. These cages let them measure everything, activity, breathing, etc.
Oh, and speaking of activity, figure 2C confirms they weren't just running around more, right?
Correct. Locomotion, general movement, pretty similar across the groups. So the weight loss isn't because they suddenly became marathon runners.
Okay, ruled that out. What about how they were getting energy? Figure 2 looks at RER.
Respiratory exchange ratio. It basically tells you if the body's burning more carbs or more fat. And for the CC, mice missing cysteine, the RER went down. That means they shifted towards burning more fat.
Burning fat, which aligns with losing weight.
Exactly. And the DEXA scans in 2E confirmed it directly, a really big drop in their body fat percentage.
And the pictures in 2F and 2G are pretty stark, aren't they? Looking at the fat tissue itself.
They really are. Those are microscope images of white adipose tissue, the main fat storage depots. In the cysteine-deprived mice, the fat cells are they look almost completely emptied out by day 7, just drained of fat.
Well, but importantly, the cells weren't dying off in large numbers.
No, they checked for markers of cell death, like caspase-3, and didn't see significant signs. So it's fat loss, not tissue destruction.
That's good. And then 2F shows something called browning.
Yeah, this is fascinating. White fat cells started looking and acting a bit more like brown or beige fat cells. Those types of fat cells are specialized to burn energy for heat, not just store it. The UCP1 staining indicates the switch.
And this happened faster and was more pronounced than just with calorie restriction.
That's what they report, yeah. A rapid and significant browning effect. They also mentioned in the extended data that visceral fat, the really unhealthy kind around organs, was lost quickly too, while muscle and liver tissue looked mostly unchanged.
So big picture from figure 2. Less eating, yes, but also a major shift to fat burning, fat cell emptying, and even turning some storage fat into energy-burning fat.
You got it. It's a profound metabolic reprogramming triggered by lacking cysteine.
All right, so figure 3 delves into the why at the molecular level, what genes are being turned on or off.
Yep, RNA sequencing time. Figure 3A shows the design. They looked at liver, muscle, and fat tissue. And importantly, they compared the cysteine-deficient group to a tryptophan-deficient group.
Using tryptophan deficiency as a control for general amino acid stress.
Kind of, yeah. To see what changes were specific to cysteine. So figures 3B and 3C focus on the liver, major metabolic control center. They saw increased activity upregulation of genes involved in stress responses, specifically the integrated stress response, or ISR, and the oxidative stress response, OSR.
Stress responses, okay. Things like FGF21 and GDF15.
Those were 2 key ones, yeah. Hormones known to influence metabolism and appetite under stress. And conversely, they saw downregulation, decreased activity of genes needed for making cholesterol and fatty acids.
So the liver is like, okay, stressful situation, stop making fat, signal the stress.
Basically. And while tryptophan deficiency also triggered some similar changes, the effects were way stronger with cysteine deprivation. They even suggest there might be some synergy happening between the ISR and OSR pathways in the liver.
Interesting. What about in the fat tissue itself, figures 3 and 3E?
Similar theme, but different details. They saw suppression of a key gene called SREBF1 and its targets. SREBF1 is like a master switch for fat production.
To turn off the fat-making machinery in the fat cells, too.
Exactly. Plus, they saw increased expression of genes that break down fats and that mild increase in UCP1 again, linking back to the browning effect.
And muscle. You mentioned that was a bit different.
Yeah, the extended data showed muscle mainly ramped up the oxidative stress response genes, but not so much the integrated stress response. The idea is maybe muscle has such high protein levels normally that it doesn't sense the amino acid drop quite as critically for the ISR.
Huh. So different tissues sense and react slightly differently. Makes sense.
It highlights the complexity. Now, Figure 4 provides more direct biochemical proof for these stress responses. Figures 4A and 4B directly measure glutathione GSH. And sure enough, in the liver and muscle of the cysteine-deprived CC mice, GSH levels plummeted.
Which makes sense, right?
Yeah.
Cysteine is needed to make GSH. Low cysteine, low GSH.
Precisely. And that drop in GSH directly connects to oxidative stress. They also showed evidence for OSR activation, the NRF2 protein moving into the cell nucleus and more NQ01 protein being made in the liver.
Okay, so OSR activation confirmed. What about the ISR?
Figure 4C shows proof for that too. Increased levels of a phosphorylated form of EIF2, that's a classic marker of ISR activation in the livers of the cysteine-deficient mice. They also mentioned having trouble creating mice that lacked both CSC and GCN2, a key ISR sensor. That hints that maybe the ISR is actually crucial for survival when cysteine is low, perhaps even active at a low level normally in these CC mice.
Interesting. And the stress hormones?
Figures 4 and 4E show big increases in GD15 and FGF21 circulating in the blood, matching the gene data. And this wasn't just in the CC mice. Normal mice deprived of both methionine and cysteine also showed this hormone surge.
So these hormones are likely mediating some of the effects, like reduced appetite.
Very likely, yeah. And consistent with shutting down fat production, figure 4 shows lower levels of the ACC1 protein, which is involved in fatty acid synthesis.
And less fat synthesis means less fat in the blood.
You'd expect that, and figures 4G and 4A confirm it. Significant drops in serum triglycerides and free fatty acids in the cysteine-deficient mice.
Okay, so the picture's coming together. Cysteine loss, GSH loss, oxidative stress, plus integrated stress hormone changes, less fat production, fat burning. But is that the whole story?
Great question. That's what they tackled starting in figure 4I. They tried to isolate the role of GSH depletion using that inhibitor, BSO, but in tryptophan-deficient mice this time.
Ah, so induced GSH depletion without the cysteine deficiency itself.
Right. And while giving BSO did cause some weight loss, it wasn't nearly as much as with cysteine deficiency alone.
Suggesting GSH depletion is part of the story, but not all of it.
Exactly. Figures 4J and 4K compare the liver gene expression. OSR activation was similar between the cysteine-deficient and the BSO-treated tryptophan-deficient mice. ISR was maybe slightly weaker with BSO. Fat synthesis genes were down in both.
But the weight loss is different.
Right. And figures 4 and 4 show that the biochemical markers, like PEIF2A for ISR and the low GSH levels, were actually pretty comparable between the two groups. ACC1 protein and GDF15 hormone levels were also similar, figures 4N and 4O, yet the weight loss difference persisted.
So, if it's not just GSH depletion and the resulting stress responses, what else is cysteine crucial for?
And that, as summarized in 4P, leads them to the other major role of cysteine we mentioned earlier, making coenzyme A.
Co-A. And that's where figure 5 comes in.
Exactly. This was, I think, the most surprising part of the study. Figure 5A reminds us of CoA's structure and how it's made needing both cysteine and vitamin B5. Then figures 5B, C, and D deliver the punchline. CoA levels dramatically decreased in the liver and also somewhat in muscle of the cysteine-deprived CC mice. And this depletion got worse over time.
Wow. So cysteine limitation directly causes CoA depletion. That wasn't necessarily expected.
Not to this extent, no. CoA is involved in so many core reactions, it was thought to be more stable. This suggests it's actually quite sensitive to cysteine supply, at least under these conditions.
So what happens when CoA levels drop?
Well, CoA is essential for feeding fuel into the TCA cycle, the main energy-generating hub in our mitochondria. Figures 5E and 5 show the consequences. They looked at metabolites. In the urine, things like pyruvate, citrate, bacchidoglutarate were being excreted much more. That suggests a bottleneck in the TCA cycle because there isn't enough CoA to process them.
Like a traffic jam in the metabolic highway.
Analogy. And inside the liver cells, intermediates from glycolysis, the pathway before the TCA cycle, were piling up, suggesting a shift towards less efficient anaerobic metabolism, perhaps.
And figure 5G provides functional proof of this mitochondrial problem.
Yes. The cysteine-deficient mice consumed less oxygen, which mitochondria need, and crucially, they couldn't survive when fed only galactose, a sugar that forces reliance on efficient mitochondrial energy production, oxidative phosphorylation. Control mice, or tryptophan-deficient mice, were fine on galactose, but the cysteine-deficient ones weren't. Strong evidence their mitochondrial powerhouses were compromised, likely due to low CoA.
That's compelling. And they ruled out vitamin B5 being the primary issue.
They did. Figure 5 shows that just making mice deficient in B5 didn't cause weight loss. And removing B5 in addition to cysteine didn't make the weight loss worse. So cysteine was the real limiting factor for CoA here.
Okay. But there's a twist with B5 loss. Figures 5I and 5J.
A potential feedback loop, yeah. They found increased activity of an enzyme called VNN1, or pentethinase, in fat and muscle. This enzyme breaks down a B5 precursor. This could potentially lead to more B5 being lost in the urine, making it even harder to rebuild CoA levels once cysteine is scarce.
Sort of kicking the system while it's down.
Kind of. Figure 5 clay just neatly summarizes how you'd expect levels of cysteine, GSH, and CoA to change under these different conditions, highlighting the CoA drop with cysteine deficiency.
And the final experiments really nail the CoA connection, don't they? Figures 5 out to 5N.
They do. When they stopped the cysteine deprivation, just giving back cysteine wasn't enough for the mice to fully regain weight if vitamin B5 was also kept low. Their CoA levels stayed low too.
But if you gave back both cysteine and B5,
Then CoA levels recovered better, and the mice regained weight properly, showing both are needed to replenish that critical CoA pool.
okay, so figure 5 puts it all together in one model.
Yeah, the grand synthesis. Cysteine deprivation hits two key things: glutathione, GSH, and coenzyme A, CoA. The drop in both triggers stress responses, OSR and ISR, leading to hormonal changes and reduced fat synthesis. Critically, the CoA depletion impairs mitochondrial function, reducing metabolic efficiency, causing fat to be burned or lost, and metabolic intermediates to be excreted. It's a multi-pronged hit.
Wow. That's a really elegant unraveling of a complex process.
Mhm.
So thinking about the innovation here.
Well, I think the systematic approach itself was innovative, actually comparing the removal of all those amino acids side by side and combining the genetic knockout, CC mice, with the dietary restriction gives really strong evidence. Plus, integrating all the different data types, weight, food intake, metabolism, gene expression, metabolites, proteins, that multi-omics approach is powerful.
And identifying CoA depletion as such a major, maybe even the major downstream factor, that seems like a real breakthrough.
Absolutely. It wasn't the obvious first guess. It adds a whole new layer to how we think cysteine impacts metabolism.
And this definitely provides a new perspective on weight control, doesn't it?
It really does. The idea that targeting the availability of just one specific amino acid could be such a potent and importantly reversible way to influence body weight is, well, it's provocative.
That reversibility seems key. You could imagine, way down the line, carefully controlled dietary phases for metabolic health, maybe.
It opens that door for exploration, certainly. The rapid, reversible nature in mice is striking. But, the standard disclaimer applies. Translating this safely and effectively to humans would require a lot more research. Rigorous clinical trials are essential.
Of course, safety first, but the potential is intriguing.
And it might have implications beyond weight loss, right? Like in cancer metabolism, where some cancers are known to be particularly reliant on cysteine.
That's a good point. This cysteine GSH CoA axis could potentially be a target in diseases where you want to induce rapid metabolic shifts.
Yeah. And just the experimental strategy itself, this combination of genetics, precise nutrient manipulation, and deep metabolic profiling is a great model for studying other nutrients too. How do other vitamins or amino acids exert their effects? This gives us a template.
It definitely inspires looking deeper into the connections between what we eat, our cellular stress levels, and overall metabolic function. So pulling this all together for you listening, this deep dive really shows how one single amino acid, cysteine, can pull some major levers in your body's metabolism and weight regulation. It's a fantastic reminder that biology is incredibly intricate. Small changes can cascade into really significant effects.
And I think what's truly exciting here is the glimpse it offers into potential future strategies. The possibility of precisely tuning metabolic health, maybe even body weight, by carefully managing specific nutrients like cysteine. And again, that reversibility is a huge factor, potentially offering a more adaptable approach than some current, more drastic diets.
Yeah, definitely food for thought. Okay, so here's a final idea to chew on. This study really forces us to reconsider how stable we assume certain cellular components are. We thought of CoA as this steady workhorse molecule.
Right, just always there, doing its job.
But this shows it can be surprisingly sensitive to the supply of just one precursor, cysteine. It makes you wonder, what other key metabolic nodes, what other molecules we think of as stable, might actually be dynamically regulated by nutrient availability in ways we haven't fully appreciated yet.
That's a great point. Uncovering those hidden dynamic points could unlock completely new approaches for understanding and treating metabolic diseases down the road.